colega, un científico sumamente reconocido mundialmente en la bioinformática. Entonces cuenta con más que 75,000 citas en Scholar Google y con un índice H de 113. Entonces Peter es también un gran amigo y colega mío desde hace muchos años y un gran aficionado de la cocina mexicana y la mayoría de las cosas mexicanas. Entonces, normalmente tenemos el gran gusto poder recibirle aquí cada año. Entonces, desafortunadamente con la pandemia había 2 años donde no era posible, pero qué bueno que ya estamos ya sueltos para viajar de nuevo. Y va a estar con nosotros en el C3 hasta el martes, donde después va a ir a Guanajuato y las colaboraciones que tiene ahí con gente en siempre estaba en Irapuato, ¿no? El punto es que Peter tiene un gran espectro de intereses en la evolución, la bioquímica y yo había dicho que a lo mejor tratar de darnos una plática bastante general hoy, entonces, y va a hablar bueno, esencialmente modos de cambio evolutivo en biomolecules, ¿no? Gracias, Peter, por estar con nosotros.
Thank you. Okay, so this works. Thanks a lot for the introduction and for the opportunity to speak here. It's a great pleasure to be back here and visiting Mexico again. What I want to talk about today is a somewhat esoteric mode of evolutionary change that we stumbled across in our work on consensus structure. And sort of by way of introduction, I just want to give a very brief overview on how we view models of evolution of biomolecular structures. The basic idea is that obviously variation is introduced primarily at the level of a genotype, so just by genetic mutations. And these genetic mutations then translate into modifications of a phenotype of a structure. And then this structure interacts with its environment, and you can measure all sorts of parameters out of that structure that eventually relate to fitness. So the interesting thing about the relationship of sequences and structures in biomolecules, both RNAs and proteins, is actually that you have a very redundant a map from the genotypes to the phenotypes. That is, a lot of the different genotypes, a lot of mutants actually do not change appreciably the phenotype. I mean, there make slight differences because you have a different sequence, of course, so it's not exactly the same, but at a reasonable level of course graining that you can use to get explanations, you can essentially say, okay, so there's millions, billions, trillions of sequences that all will fold into, say, a tRNA structure that actually functions as a tRNA. They all have the same sort of clover-leafy structure. They all get bound by the same binding partners, processing enzymes, and so on and so forth. And what you're interested in essentially is how does this redundancy of sequences and structures change?
So a question here, Peter, is so on shape space, there's obviously key template structures, right? But what I want to say is that obviously with a linear sequence, then it's easy to give a complete specification of the entire configuration space, right? Just one after another, you just say what it is. But one of the things with phenotypes for me is, well, what the hell do you put in there to say that's a degree of freedom versus that isn't? So obviously, if you think of true phenotype like us, then forget it, right? You know, what is the shape of your eyebrow or something like that? What is it in shape space?
So if you go to much more complex phenotypes, essentially you have to go to some type of feature-based description, and this will never be complete. But in many circumstances, you will at least know what are the, or you can at least in principle know what are the dominating contributors. So what are the features that really matter for a particular aspect that you particularly care about. The advantage that we have in using things like molecules as models of phenotypes is that there we understand actually what in reality is developmental biology and interaction with the environment and whatnot, because in the molecular case, it's really just physics. It's really just the folding up of the molecule in a particular environment. And if you look at these tRNAs, I mean, they look like very complicated molecules, but if you go a step back, abstractly, they actually look very simple. What you have is essentially something like this. You have helical paired regions where the fact that things base pair in a complementary manner already implies that the three-dimensional structure is helical. And then you have essentially flexible loops in between that you can't really see that easily here. This is all these areas where you don't have these nice parallel base pairs. And what happens on top of these secondary structure formation is that, of course, you fold the whole thing or you embed the whole thing in a three-dimensional shape. However, in the case of RNAs, what makes RNA such a nice model to work with, also just as a model to explain a phenomena, is that these secondary structures are very much a real thing in RNAs. First of all, they are folding intermediates. So the way how the structure is formed in reality is that actually you first form something like this with the loop still being flexible, and then it kind of folds up in 3D and locks into a three-dimensional structure by forming extra interactions between mostly the loop regions. And the second thing is that the stacking of the base pairs and the formation of these loops accounts to something like 95% of the thermodynamic energy of folding. So you can, the secondary structures come very close to actually modeling the physics of the folding process. And the third thing is that for the secondary structures, which are just combinatorial structures, so they're completely discrete models. So this will basically be the only 3D picture that I'm showing you for more than illustrative purposes. We never essentially work with three-dimensional structures. We just work with these two-dimensional combinatorial abstractions. And they have a very simple structure. They consist of a bunch of vertices. There is a rule that says which bases can pair. So you have Watson-Crick, so GC and AU base pairs. And in addition, you have the wobble base pair GU in RNA. And you can, of course, revert them in the order of the sequence. So there's out of the 16 combinations of nucleotides that you can have, there is 6 legal base pairs that can be formed. And essentially everything is to a good approximation you can think of as a loop or as an unpaired position. And then we have this no crossing rule that basically says base pairs don't cross each other. So if you have a base pair from I to J, there is no other base pair that connects the inside of that base pair to the outside. And this non-crossing rule gives us a very simple recursive structure on these structures. So essentially you can think of every structure, and I think I have that on the next slide actually. You can think of these structures in terms just of matched up parentheses. So this is a very simple representation of structures that we're using, and this structure, simple representation of base pairs actually being just matching parentheses or The way how you read this, right? This is a hairpin loop that has 4 unpaired bases in the middle. Then you have a base pair that encloses these 4 base pairs, and then you extend that stem. So our tRNA clover leaf structure looks like this, right? So you have 1, 2, 3 hairpin structures inside, little stems with loops on top of it, and then you have a base pair on the outside that kind of hosts these 3 stems. And what you can use that for is a very simple recursive algorithm to calculate energies, to calculate the optimal structures in there. And in a nutshell, the idea is any structure on some subsequence can be written as either an unpaired base and a shorter substructure, or you have a base pair from the first position to somewhere else in that substructure, and that base pair, by the non-crossing rule, divides up the structure into 2 independent structures, one inside and one outside. And that immediately gives you a recursive way of calculating these things. Yes?
Yeah, just very simple, very short question. This way of looking at it is applicable to other structures other than tRNAs, or it's particularly useful for tRNAs?
No, this is a general building principle for the structure formation in RNAs in general. So no matter what, ribosomal RNAs also adhere to that.
Okay.
This is an approximation, so you sometimes can have this type of crossing base pairs, but very good approximation, you can actually negate it or add it as an add-on tertiary contacts. The same rules also apply if you want to work with single-stranded DNA, but single-stranded DNA is not a very common phenomenon in nature. There are single-stranded DNA viruses where this becomes important, and so we can, and also our software can handle also the DNA case, but that's just much less important in terms of application. And for double-stranded DNA, this is not interesting really, because there you have just one single very long helical stem regions. For DNA aptamers, of course, it's then important. It's important for artificially designed DNA structures. So these kind of methods are also used, for instance, in all these applications to DNA computing, where you have interacting DNA, and that you usually do in DNA just because of chemical stability. You don't implement that in RNA. So the general rule is everybody can work in a biochemistry lab with proteins, but with RNAs, you really have to know what you're doing, and you have to work very, very cleanly.
So basically, there's a small set of building blocks above the nucleotides themselves, right?
Yes.
And it's a hairpin loop or a strand or whatever it is.
So it actually comes at the next slide.
Okay.
So there is this. What you have in practice is actually, it's not the single base pairs that contribute to the energy, but you actually have the stacking of base pairs that contributes stabilizing contribution to the energies by the stacking of the pi electron clouds of the plain nucleobases. So this is kind of a purely chemical, if you want, contribution. And then all the loops essentially are destabilizing by means of an entropic contribution that you can very easily explain. The reference state is just a random coil state. And in a loop, you have the ends of that random coil constrained to being a base pair. And that reduces the conformational degrees of freedom, and therefore reduces the entropy. And by delta G being delta H minus T delta S, that gives you a destabilizing contribution to the free energy of folding. So you have these sort of counteracting effects from the base pairing and from the loop. So in the realistic case, you also can get these dynamic programming type solutions. So I like to write these in these hieroglyphics. that basically says, okay, a structure is a structure that begins with an unpaired base or with a base pair. And if I have a structure that's enclosed with a base pair, that's either a hairpin or an interior loop or a branching loop. And then for the branching loops, we have a certain decomposition that makes that easy. And essentially, it's this recursive structure that I'm writing down here that you can think of as a grammar if you want, or just as a set of 3 recursive equations where these are all the indices that appear in there. This is essentially the whole algorithmic core that's underneath RNA folding. And the nice thing what you can do with that is you not only can calculate the most stable structure, you can also calculate alternative structures. You can calculate the whole partition function for a given structure. So you have essentially complete access to whatever you want to know. You can calculate melting energies and whatnot for these molecules. What we can also do is we can ask, so now if we take the ground state structure, if we take the most stable structure as an approximation for the structure that we're interested in, rather than the whole ensemble, just to make the arguments on the presentation a little bit easier, we can actually ask how easy is it to go from one structure to another structure. And the way how you do that numerically is essentially you just calculate for a large number of sequences what are their secondary structure, and then you make point mutations into these sequences and ask what are the ground state structures of those. And what you find is, first of all, there is a relatively small number of frequent structures, so structures that are relatively easily accessible, that easily form from sequences. And essentially you get this kind of Zipf's law type power law distribution that even has a big hump at the beginning for frequent structures. And it's essentially these frequent structures sort of from somewhere here in the middle, and not this tail of very rare structures that matters for evolution, because this stuff down here is essentially for longer sequences is inaccessible. It's extremely unlikely that you will ever stumble across it. And whatever it does, it's basically the unique structure. So there's one sequence with one structure and there's nothing around it where you can elaborate on it. So those things are essentially evolutionary non-interesting. The interesting part is actually the kind of phenotypes that you can get recurrently that are stable against mutations, that have many realizations where you can get variations on the theme to optimize sort of bells and whistles in evolution. So essentially, if we think evolution, we only have to care about the frequent things that nature also can easily find through an evolutionary search process. And if you only look at these frequent structures, what we realize in RNA and also in proteins for that matter, is that they form very extensive what we call neutral networks, not to be confused with neural networks, so no machine learning here. Neutral network simply means neutral in the sense that a point mutation, so going to neighbors in that network doesn't change the structure, and network just because it forms an essentially connected graph.
So is it an assumption, or it's really
this is
no? What I was going to say, assumption is that the move set is point mutation versus no. Observationally, you never see anything else.
So I mean, you would also see something else. You can make more than one point mutations, then you get a little bit further, but that doesn't qualitatively change the picture.
But you don't get cleaving of the macromolecules.
No.
You know, it's not like there's no recombination type.
So I mean, if you do recombination, remember, if you do recombination between things that are very similar, then the recombinants kind of look like mutants. So we haven't looked at what the recombination. We haven't looked much at what this recombination landscape, sort of this space of recombination. But
as an experimental phenomenon, do you see recombination type events?
You do see that in some RNA viruses, for instance, and also in, but not in general. So I mean, if stuff is encoded in DNA, RNA genes are very small, so it's not very likely that you see a recombination event in the middle of a tRNA. But this is kind of a James Bond type of business here. So never say never. Whenever you can think of it, nature probably did it at some point. So I'm sure there is also some recombinant non-coding RNA somewhere, but it's just not a frequent process. And what that does is that essentially you can move around on this sequence space and not change the structure or not change the structure appreciably. And most or many of the RNA molecules use their three-dimensional structure and therefore also their secondary structure to exert their function. So what it does is it says, okay, there is a selection pressure to conserve the structure. There will also be a selection pressure on conserving some elements or some features of the sequence. But for much of the structure, the sequence really doesn't matter, right? So we get variability in the sequence and conservation in the structure. And this is a rather nasty, complicated example. It's the so-called U7 sNRNA. It's a small RNA gene that's relevant for the processing of the histone genes, which is kind of a core type of proteins that makes up the chromatin and kind of protects our DNA and is important for epigenetic control. And these genes in the replication-dependent ones of them, they don't have a normal polyadenylation mechanism for the mRNAs. They have a different way of processing the mRNAs. And this weird RNA is involved in that. And I'm showing that basically for two reasons. So first of all, there is a bunch of conserved sequence elements in there that we do see in these non-coding RNAs, and but there is also this conservation of essentially a hairpin, and this hairpin is important because that's kind of what's used to determine where the end of the whole molecule is, and eventually also where the histone is cut. And there is a lot of variability in there, both in terms of the sequence that is sitting there and also in the size and details of that structure. So also these structures are not always perfectly conserved. And the same thing, and I'm only mentioning this for time reasons very briefly, so you can make the same arguments with sort of much less with much less systematically acquired data. You can make the same argument for proteins. So for proteins, you also have a conservation of structures on top of sequences. This extensive network of protein sequences that essentially build the same structure, which, by the way, is essentially an effect that's heavily used in recent advances in machine learning-based protein folding. So AlphaFold wouldn't work if this would not be true, because AlphaFold actually recognizes that a protein fits into an evolved class of proteins with particular structural features, and it actually capitalizes on the fact that this is well-established kind of local structural conservation that can be derived from the sequence because the machine local sequences. So we get these consensus structures, and the normal situation is that you have your sequences, columns in a sequence alignment are homologous sequence positions, so homology defined now in the strict evolutionary sense as by common descent. So the interpretation of a sequence alignment, at least if it's an alignment that comes out of phylogenetic comparison, is that sequence positions in the same, so entries in the same column actually derive from the same ancestral amino acids, so really are kind of the same amino acid in different species, in different sequences. And from this multiple sequence alignment, you can infer where are the contacts between these columns. And what you observe normally is that homologous columns will make contacts that are preserved in the consensus structure. So if you preserve a structure, you preserve the structure by preserving individual contacts, by preserving individual local neighborhoods in the structure. This is the usual type of consensus structures that we know how to detect and that we know how to evaluate, basically by starting from the sequence alignment, looking at variations between columns in this sequence alignment, is essentially inferring with more or less sophisticated statistics on where are the evolutionary conserved contacts. In proteins, you can do that. You just need an awful lot of data. So there's very nice work by Chris Sander and David Marks, who showed that you can do this for large, if you have very large alignments of hundreds or thousands of related protein sequences. This works very nicely, that you can get contact matrices out of it, and the contact matrices actually give enough constraints essentially to also compute real three-dimensional folds out of that. For RNAs, we are in the lucky situation that because base pairs have only one pair, so a base can only have one or no pairing partner, it's much easier to identify the paired sequences. And we have also methods of combining sequence covariation and consensus and thermodynamics driven structure prediction. Essentially, what you can do is you can take the whole RNA alignment and essentially put that into the folding algorithm and just put in as an extra rule only columns can pair that can pair in almost every row, right? So you have to have a TA pair or an AT pair here, or a GC or a GC or whatever. So one of the 6 pairs out of the 16 possible in order to make this pair. And if you enforce that for every column, for every row, or for almost every row in a pair of columns, you get a very restrictive set of pairs of positions that can even form a base pair. And if you then throw that into a thermodynamic folding algorithm, it gives you already with something like 10 sequences a very good prediction of what the consensus structure will be. So for RNAs, consensus structure prediction is just a lot easier than you can than for proteins. But in principle, it's the same thing. So this is this RNA RE4 program that does that. I don't want to belabor that. So if selection acts to preserve function, and function is closely tied to structure, then this implies that we get selection that preserves structure, and the primary mode of preserving structure is simply preserving local contacts. And therefore we get consensus structures, and these consensus structures means that structurally corresponding contacts are formed by homologous amino acids or nucleic acids. And this is what we can very easily, or relatively easily as bioinformaticians, detect in real data. So this is kind of the normal thing that happens. But first of all, the idea that we have just one secondary structure is a rather crude approximation. Same thing as that the idea that there is in detail only one structure in a protein. So essentially all proteins have multiple structures. Sometimes they need this allosteric or whatever to exert their function. But even if this is not true, the interactions between individual amino acids or between individual nucleotides not strong enough to really lock that into one frozen state, but rather you get an ensemble of different structures. This is just one example. What you see on top here is just some RNA sequence, whatever it is. And then you have down there a number of alternative structures. And what you see here is the computed energies for folding that. So free energy is expressed in kilocalories per mole, and thermal energy is about 0.6 kilocalories per mole. So this is about so this range is about 1.5 times the range of thermal energy. So this is the highly populated part of that structure. So in reality, you get a mix of all these structures. And what you can see is, well, there are some base pairs that are essentially always there. And then there is a bunch of other base pairs that are sometimes closed and sometimes not.
But now you have to look at the possibility of neutral networks at that level, right?
At that level,
Yeah.
and essentially, so then neutrality becomes a matter of degree. You would basically say, okay, so most structures are very similar, or you have a very large overlap in the base pairs that are almost always there. So one way of thinking about that is to abstract these ensembles by their what's called centroid structures, which is so for every column, you ask, is this column paired at least 50% with a fixed base pairing partner? Then I only take these base pairs into account and assume that everything else is flexible or unpaired. So we can think of three states: predominantly paired, predominantly unpaired, and some flexible stuff in between. And only compare these patterns, for instance. So it becomes a matter of what exactly you want as your definition.
So the problem then is like, what energy resolution do you have in your model?
It always is. All these things, and this is really a point to emphasize. Whenever you compare molecular structures from two different molecules, so a wild type and a mutant, the structures by definition at the atomic level are different because they are actually different molecules, right? So when you say this is the same structure, you're implying that you're looking at it at an appropriate, whatever that means, level of abstraction. And here our level of abstraction is secondary structures, or a little bit cruder than secondary structures.
Mm-hmm.
Mm-hmm. So this, I think, for the sake of time, I don't really want to go into that. This was basically just a way of saying, if we have these ensembles of secondary structures, there is also ways and means of actually assessing that experimentally. You do this by methods called chemical probing, where either you modify the RNA chemically by just binding some organic residues to the individual nucleotide in a way that depends on the structure. So there's plenty of reagents that will attack flexible, therefore unpaired regions, and then you basically detect by means of a deep sequencing technique where these structures go. So this is done with a thing called shape analysis, or we have our own protocol that we developed just very recently with the biochemistry group in Leipzig, where you can. So I'm not going into any of the biochemistry. So the whole take-home message of that is, normally what people do is you do this chemical modification, you cut up your structure, and then you measure one side of the RNA. And this is basically a clever way of measuring both sides of the cut, and therefore getting much cleaner data out of it. This shows that you actually get signals out of this. This is maybe interesting, sort of a very generic way of using this type of experimental data in the RNA structure prediction. So what the information that you get out of these experiments is, this sequence position is likely unpaired, or I don't know anything about it because I don't get a signal. What you don't get out of any of these methods is actually a direct measurement of the structure. So it's a very indirect way of assessing structures, and you still need to predict what the correct structure is. And one way of doing that is to take this external energy, this external data, so that basically convert the signal that you see in a probability that a particular base is unpaired in this case, and then convert that into an energy contribution that's basically minus RT times the log odds ratio, so minus RT times the log odds ratio of that thing to be unpaired, and then you add that as an energy contribution, as a bonus to the folding energies, and this works actually very, very nicely. And I don't want to belabor that. there is a natural way of hairpins, a natural way of regulating genes by this type of transcriptional hairpins, that transcriptional riboswitches that appears quite frequently in bacteria, where you have an aptamer part that binds some ligand, and depending on that ligand, the terminator is formed or is prohibited. And sort of the conceptually more difficult case is to say, okay, so I want a transcriptional on switch. I want a structure that if the ligand is not there, I form this terminator hairpin, and if I have the ligand, then it binds in a binding pocket in this RNA structure, changes the RNA secondary structure in such a way that the terminator cannot be formed anymore, and then transcription goes on. And the fun part is you can actually design this on the computer. We built about 10 constructs, sent them to our biochemist friends, and 5 of them worked out of the box. So, and I guess 1 out of 2 for this kind of complicated system is reasonably good as a rational design. So at least they were very happy that they didn't have to go through hundreds of examples.
Who models the interaction between the ligand and the?
So this we took from the literature. There is so you can in principle use docking calculations, but in practice we just don't know enough about RNA ligand interactions also energetically. So in reality, the software is not good enough to really do that from scratch. So, but you use aptamers. So right. But there's very large libraries of aptamers that were produced by. So this, yeah. So the aptamer sequence is something we took out of the catalogs of experimentally produced aptamers. So it was known that there is an aptamer that binds theophylline very specifically with roughly whatever binding energy. And then the task was, okay, so now we know an aptamer that binds theophylline, use that to build a full expression platform for a riboswitch that acts as a transcriptional on switch. And you can essentially replace, and we did this for a few other examples, you can replace this theophylline aptamer by some aptamer for something else that you're interested in. So this, again, just sort of in order to say that you can use these secondary structure models really in a lot of detail to understand something up to the point that the model is actually accurate enough to make predictions that you can use for design that actually works then if you build it into an organism, in this case, E. coli. So we've seen that there is all this structural variability. We've also seen how we get consensus structures as the kind of congruent mode of evolution where sequence and structure evolution fits together. So the question was, what is if this is not the case? Is this actually conceivable that you conserve structure in such a way that the conserved structure now is not formed by homologous contacts, so that you preserve the shape of the molecule in such a way that the individual contacts are actually changed? And not just one contact is changed, but essentially more or less all the important contacts are changed. So the idea is essentially if function is only cares about local structure, then there is no reason to preserve this global homology of every column. We could actually just shift the structure over, and looking for this was really motivated because if you look at alignment, say in the RFAM database, which collects consensus structures for RNAs, you sometimes have the impression, well, this makes very nicely preserves the structure, but if I look at the alignment, this alignment doesn't look right. This looks shifted. And the question was, how can we go after that? So can there be incongruences between the conservation of sequence and structure, in the sense that we conserve sequence and we conserve structure, but we do not conserve the lineup between sequence and structure? And this is just to show that one can build this. This is a made-up example of just from a secondary structure prediction of a protein. And to just show you how easy it is to do this, right? So you can actually take this alpha helix here, and we just make a couple of with 4 amino acid changes. You can actually move this alpha helix and this beta sheet over by 1 in the structure. So every contact in this structure is actually changed on the 3-dimensional level. Nevertheless, you have sequences that they have the same lengths. There is 4-point mutations out of some 60 amino acids or something like this. So this is a perfect alignment. Everybody will believe that those are the homologous positions. But if you align this thing on structure, you get these very large stretches where sequence and structure, where the sequence doesn't fit anymore, right? And so what we were basically thinking about, okay, how do we represent this type of situation? And the way how you represent this type of situation, essentially, it's okay, so there is shifts between sequence and structure at these two positions. And how do you detect that, or how do you model that? That is basically the question that I want to answer in the rest of my talk. And Chris basically can tell me whenever to shut up, because after the next few slides, it becomes a little bit bioinformatics-y, and if this is too specialized, we can also just stop there. So the basic idea how that works in evolution. So how do you get that? So here I've written that in a very simple RNA example. We have 2 hairpin structures. Both are hairpins of length 4. Both have very similar sequences. If we align the sequences, then what we get is these 2 sequences. And if you look carefully at that thing, the hairpins are always between non-aligned, or the base pairs are essentially mostly again between non-fitting structures, or we can make the structure fit, but then the sequence doesn't look fitting anymore in this part. And the way how you get that is essentially through an intermediate, and that's the reason why I was talking about this multiple structures that you can get from the same sequence. So again, a made-up example for simplicity. What you can say, we start with this sequence, with that structure in evolution. Then we build this intermediate, which is just a point mutation outside actually the paired structure. Now this thing here actually makes 2 structures, namely the original structure of the original sequence before the point mutation, and the structure that's shifted over by 1 with almost exactly the same energy. And if you make another point mutation into that thing, you get that structure. So if here both of these structures are essentially functional, then the next essentially neutral point mutation can either go fix this structure or fix that structure. So you can get these structural shifts without any loss of function mutation in between. So basically it becomes a question. So this is essentially a completely neutral mechanism that will give you this incongruent type of divergence between sequence and structure. This is only a small fraction of the point mutations that you can make that will preserve the structure, but they do exist, and these things actually also do show up in structure. And this is again an example. It's very easy to build rather elaborate examples where you can have very few point mutations making very large differences, very large shifts as well. So the real question now is, does that actually also occur in nature? Is this a mode of evolution that we have to think about how this works and whether this where this occurs in nature? And the point is, one can actually find this also in nature. This is a human cytochrome protein that actually shows exactly this type of incongruent shift. So it preserves the secondary structure, also essentially the tRNA structure, the three-dimensional structure on this whole protein, but it actually has this helical region that in human, compared to mammalian ancestors, is actually shifted over. And this would show up as a major evolutionary change if you sort of naively try to analyze the data, but apparently this doesn't do anything, so there's no functional difference. So this is one of the few examples in proteins that are well described, but people never looked for this in proteins, and it's difficult to do. In RNAs, we also find it, and this is just an example from a microRNA family. So microRNA 30 comes in two paralogous groups, microRNA 30A and the microRNA 30B. And if you look at their alignments, this has a very clear picture of having a structure between these two paralogs that is shifted over, where essentially the whole hairpin and therefore also the sequence of the mature microRNA is actually different. It's shifted, it's elongated on one side and shortened on the other side. So it also acquires a different seed sequence. So how do we look at that from a bioinformatics perspective? The idea essentially is, well, we have an alignment between the sequences that we started with, but we can also think of an alignment of the structures where we now make homologous positions that make up corresponding contacts, form the alignment columns. So this doesn't have a direct interpretation in terms of sequence evolution, but it has a direct interpretation in terms of what are the corresponding places in the three-dimensional or two-dimensional structure. And so now we have these two alignments, and these two alignments don't fit on top of each other anymore. So we can identify where these two alignments deviate from each other by just looking at the column patterns, right? So we have to have column patterns where in one alignment we have, say, a match, and in the other alignment we have an insertion or a deletion. So therefore one sequence gets shifted. So one alignment gets shifted relative to the other one. And whenever we encounter something like this, we just exclude, or we just include an extra score. We just say, okay, so if you want to do this, if you want to move sequence and structure out of sync, you have to pay for that. And that very naturally says, okay, so we should actually look at an alignment that has the first alignment on the sequences, that has the second alignment on the structure, and an alignment of the patterns on how we align the sequences and the structures, and compares just these patterns with each other. And then we have a score for the sequences, for the structures, and for these patterns. And as it turns out, well, this is just an alignment of 4 sequences simultaneously rather than an alignment of 2 sequences. So in principle, we know how to solve this structure, or how to solve this combinatorial optimization problem. And if we limit how far sequence and structure can be shifted out of sync, then this is actually also computationally reasonably feasible to compute this stuff. And this is basically how to do that. I'm not going into the algorithmic details for this audience. We did a very preliminary scan. So we're trying to build more fancy software for this topic now, where we looked at about 2,000 ARFAM families that were not too big and kind of therefore we could do the calculations rather easily. And we found among these 1,200 sequences about 700 families that have candidates for this type of shifts in it. And at the end, after sort of inspecting these things and filtering them, you still wind up with about 150 cases where we are confident that this really is one of these sequence to structure shift events. So this is relatively rare, but we also looked at this data rather conservatively. So I would think of that as a lower bound. And what it also says is that, well, in about 10% or so of the RNA families, this was something that occurred within the kind of recorded history. So it's not the dominating effect, but we didn't expect it. But it's something that we cannot ignore, and it's probably something that's in particularly important in two contexts. One context is the difference between paralogs. So whenever you form paralogous genes, because then this is a mechanism where you can have a moderate change in function that just comes from shifting over stuff while preserving the overall functionality of the molecule. And we think, and this is something that we're going to explore in the future, we think that this is also something that's in particular relevant for immune escape in viruses, because what it does implicitly, it actually means that if you preserve the structure, but you shift over the sequence, you start exposing very different sequence patterns. So it could actually be an important effect in immune escape in general. And yes, we can also do this same thing not only on the level of sequences and this type of simple models for, say, protein secondary structures or just comparing paired or unpaired positions. You can also write this whole thing down for a real RNA secondary structure model where you really take care about helices and things like this. This is basically the math behind that, also not so complicated. It's basically a simple recursion formula that for the people in the field looks like Sankov's algorithm. And this is basically how we're implementing this. So this last part is joint work with Sebastian Will in Paris and Maria Waldl in Vienna. And it's not completed because there's a very recalcitrant bug somewhere in our code in the moment. So take-home messages, mostly sequence and structure evolve congruently. So this is basically as expected. Congruent evolution is sort of by definition what leads to detectable secondary structures, but incongruent evolution of sequence and structures does occur both in RNAs and in proteins. And it appears to happen with a certain regularity in RNAs. So it's reasonably abundant that we do have to look at it. For proteins, we don't know how prevalent it is. We have a handful of examples that we kind of stumbled across by chance. So far, no systematic survey for this has been done. And basically, we've developed a way of handling this type of incongruences with a bioinformatics machinery that is this bioalignment. So this is basically this type of pairs of alignment that you tie together and score as a unit with a penalty for shifts between them. And last but not least, most of the work on the incongruent evolution was done in collaboration with Maria Wartl in Vienna and Sebastian Will at the École Polytechnique in Paris, and with Ivo Hopacker's group. So in Vienna is where we systematically develop this RNA folding software called the Vienna RNA package. And I showed you some of the actual applications of alternative structures and design based on such ideas that we did with Mario Mörl's group, the biochemistry lab in Leipzig. And of course, none of that would work without money. And the money largely for this work comes from the German Research Foundation and the Federal Ministry of Sciences. Thanks.
Voy a preguntar en un segundo. I'll ask in a second. Any more, Javier, or you have anybody has any more questions? Or
so I remember that many years ago, gene duplication was one of the leading causes of evolution. No? But there was not a mechanism to explain how gene duplication would develop newer functions. But I think that after listening to your talk, that maybe you have one mechanism. So there is gene duplication and then mutations, and then you change the structure.
So this is actually not my work, but work of David Lynch and collaborators. So what they showed rather conclusively is that there are different fates of what happens to a gene after gene duplication. Okay. So one of them is one copy just gets removed again. Mm-hmm. There's the uninteresting case, which is basically it is if it had never happened. That's actually probably the most frequent case, at least for tRNAs or small RNAs. This is what we see. It's easy to duplicate them, and most of the time, the duplicated copies just get eradicated again. The second case is that you get what's called subfunctionalization. You get actually deleterious mutations in both copies. So both copies lose part of the functionality. So therefore, you need both to together retain the original kind of polymorphic function of the original gene. And that also allows the two copies now to adapt better to the two different functions. So this is it's called subfunctionalization and kind of leads to an optimization of partial functions of the original gene. Mm-hmm. So this is one reason why particular, say, GPCR families expand very rapidly, and you get lots and lots of different paralogs of them, because they just start adapting to very specific signals, very specific interactions. And then there is also sometimes, and this may hang together with this incongruent evolution part, you get neo-functionalization where you actually one of the copies acquires new functions, mm-hmm. and this may be associated with this type of larger structural changes, but there's also other relatively rare events where structure changes and it now does something different, as in the case, for instance, of this ribozyme, right? So, I mean, mm-hmm. if you have this, say, the cleavage ribozyme, and you make a point mutation, and you get something that also has ligase activity. If this is useful, of course, this copy will take off and now do something completely different. Mm-hmm. But I think you pointed out a very interesting issue in evolutionary biology, right? So, I mean, it's relatively easy to understand what happens at the level of sequences. You can even translate it kind of into what happens at the level of structures. But what does that mean for the function, and which functions are actually adaptive? And what we think is that there is actually a lot of gene duplications, also a lot of new RNA genes, just expression of non-coding RNAs that in the beginning is essentially neutral. And then if you're lucky, and you're lucky with actually a fairly high percentage, you can use this additional copy for something in a particular context, and then it will very rapidly adapt to that. So for microRNA, we kind of know that this is the mechanism how you get new microRNA families. If you have RNA expression, forming stable hairpins is actually kind of one of the most frequent structures that happens. If you want to get a reasonably stable hairpin, that will be processed by the dicer and co-machinery. So you get a microRNA function, and this is a useful or not. And if it's useful, then it will be elaborated on. If it's detrimental, of course, it will be removed. So this is how it works.
Thank you very much.
Just a short comment. Thank you for the seminar. I enjoyed it very much. I think it's connected to something that has been in my mind for a while. Is the evolvability of different structures. Which patterns are possible or are allowed to certain sequences and structures, and how can they go to a pathway that derives a new structure or a new function. And obviously there are families that are more evolvable than other. And I don't know if you connect some of your work with that concept.
So at some level, yes, but we observed, for instance, when we looked at these neutral networks, what you find is so how evolution works on this neutral network is it's essentially like a diffusive Moran process. So you have kind of a cloud of mutants or cloud of variants that the center of mass of which essentially does a random walk on this neutral network. And then it produces mutants, some of which are on the neutral network, some of which are just mutants that kind of fall off the network, or kind of the adjacent of the neutral network. And what you can do is you can trace what are the structures that you see. And how many structures do you get that you have never seen before since you started that process? And it actually turns out you keep seeing new structures essentially until you exhaust more or less the whole sequence space. So there's a certain rate of possible innovation, of seeing new things in the adjacent possible, that just comes from diffusing along this neutral network. So from changing the sequence without actually changing structure and functionality comes the possibility to get completely new things that are not accessible, that you cannot make sort of from the original sequence. So this is also how we think adaptive processes for these RNAs work. Essentially, you get an RNA that exerts a certain function, and then you get this neutral process where you drift through sequence space, and at some point you find a mutant that structurally works a little bit better, and then that gets selected for. Usually that opens up a few more steps of selection, so you get kind of a cascade of selections that now you have a better variant. Now you really play survival of the fittest. You eradicate the structure that you had in your history, and you get the new one, and now this diffusive process starts again. And what that explains is this kind of punctuated equilibria type of behavior that you see at the level of phenotypes, where simply you have essentially no change for a long time, then you get some structural innovation, then you have again essentially no change for a long time, and then rinse and repeat. And you can explain that without going to any hopeful monsters or any other sort of rare event type explanations.
So yeah, okay.
Goodbye.
So for the thing about the gene duplication, then I mean, you didn't mention resilience, right? I mean, the point would be is that a bit like only having one hard disk, right? Meaning, okay, if something happens to your hard disk, you're screwed. So in other words, to what extent is it useful to have a backup? So the thing is that the backup isn't going to work unless there's some natural selection to say, hey, you did. So in other words, natural selection has to say stuff goes wrong now and again, and we're going to use the backup versus it never gets used, and it's going to drift away. Right?
So I mean, this is probably sort of now just from the top of my head, this is probably a question of selection strengths and effective population size. If you have a relatively small population and not too strong selection, having a backup is good because then you can survive the occasional deleterious mutation. If you are in a strong selection scenario and you have very large populations, as in bacteria, you don't really care about the backup for the individual. You just stamp out the copy that doesn't survive because there's a million other offsprings that do it right. So this to a certain extent, it's difficult to find models in evolution of evolutionary dynamics where this backup idea really makes a lot of sense.
Oh, I think I've got some. So the thing is that depends on, if you think of what's the origin of a gene family, right? So in other words, it's not dupe. That's what I'm not an expert. But things like Chagas or eukaryotic pathogens like that, I mean, the one thing that's clear is they have big gene families that are all just sitting there for like coding for the protein epitope or something like that. Yeah. So the point would be is that let's imagine you got up to 500,000 genes in the gene family, and the thing is that to actually code the protein epitope, you need I don't know 4, 8, whatever it would be, right? So the point would be is that it's like something I try to call sex with yourself, right? Meaning that if you only need 8 out of 1,000, you've got 1,000 choose 8 different possible protein epitopes without having to go, oh, I need to find a mutation, or I need to find another mutation, etc., etc. Right?
Yeah. So I mean, seeing this example or examples like an adaptive immune system, I guess are special cases in the sense that they are very different from, say, things like developmental genes. So we have 4 copies of the Hox clusters, and to a certain extent, you get this compensation of a loss of a Hox gene that in the developmental process can take over for another Hox gene. But if you look in detail, this is never sort of perfect, and in general, it leaves individuals with reduced viability. So I think you cannot explain these gene families, and the fact that there is still a kind of compensation that you can observe. You cannot explain this with actually that being sort of the evolutionary reason why you have these paralogs. You rather have these paralogs because they actually have separate specialized functions in different tissues. So the developmental processes are built in such a way that they are as resilient as possible, because they also, even if you don't do genetic perturbations, you have to compensate for mechanical perturbations and chemical perturbations. It gets cold or warm outside and whatnot. So this cannot be too brittle. And this is probably a reason why these processes sort of, try to pull in whatever they can in order to get it right. But in sort of, more complicated, complex organisms, this is this idea we have a backup in case something gets wrong to rescue the individual. I think this is sort of a very human-centric way of we would like to think that we have a backup system, but I think we don't.
Because the thing is that you remember, like when I remember that the paper, I did with where does recombination work best in the sense of a fitness landscape, right? And precisely you could see that it's not very good when you've got positive epistasis, right? Because all you're going to do is like put two things apart that should be together. But where it does work, in some sense, giving you higher effective fitness when you've got it, is when you had negative epistasis, and negative epistasis is precisely the thing in some sense of the backup, because you got an or function, not an and function, right? So as soon as you need A and B, if you cut between A and B, you're screwed, right? But if you need A or B, hey, cut them along, because I'll be better off, from that point of view. Now the thing is, it might well be that experimentally, you don't, that's where you're much more expert than me. But this thing of, so it doesn't have to. So but I guess what I'm saying, it doesn't necessarily have to be viewed as purely a backup. This Chagas example is a case of saying having multiple genes for the same thing. If your genotype to phenotype map is not one-to-one, then the point would be is that can be a big, very useful. Now, how often is it not one-to-one?
If you have a way of if you have multiple genes and you have a manner of regulating them in response to your environment, but that's not the backup function, right? That's not the way
absolutely. Absolutely.
I think I'm just very reluctant to believe this idea of having paralogous genes and so on and so forth as a backup system.
Yeah, I guess it's
because if you have a perfect backup, there is a cost to having a larger genome. So if you only need one or the other, there is an immediate benefit of getting rid of one. So you'd have to have this situation where you then damage the second one that's big enough that it matters under your selective pressure. So it gets difficult to come up with scenarios where this thing that's only there as a backup can actually be sustained over long evolution.
I think it's a case of defining well what you mean by a backup, right? Yeah. So what do I mean by that? So back to the Chagas case, right? So imagine you've got several genes that can be picked from. So in other words, the fitness is no different than imagine you just need 2 genes to code for the epitope, right? And you get 8 that can do it, right? So the thing is that if you only have 2, there's no fitness versus 8, there's no direct fitness advantage, right? But the point would be is that if in an adaptive sense, so there's no point in having a backup, right? So in other words, but in an adaptive sense, if the immune system can say, hey, now we can lock on to this pathogen, then you need to be able to go and find, hey, I need a new thing, right? So then it's a backup, but for a new evolutionary situation, right? It's not just saying backup, hey, there was a deleterious mutation. It is a backup, but not in the sense of you're thinking of it. You're thinking of it in the negative sense, like, well, what happens if shit happened? Well, but this is basically saying I can bring in a new positive adaptation because, hey, I can ask this.
So I would kind of rather say basically you have to make it, but that allows you to adapt during your lifetime.
Yeah.
To a broader range of environmental pressures.
Potential environmental pressures, right?
I would kind of not think of that as a backup.
Yeah, so but structurally, it's the same thing, right? I mean, I'm thinking of the gene duplication. It's saying, hey, it's helpful to have 8 genes that can potentially do with the same thing, where you only need 2, right?
way of recombining between three of them.
Yeah. I mean,
So something that's much simpler than our adaptive immune system is just having a bunch of genes.
Yeah.
Just don't get the diversity that we have in our adaptive immune system with some extra somatic mutation steps and whatnot.
Yeah, I mean, in that sense, it's obviously something like a pathogen like that, in the chronic disease, it's under evolutionary pressure all the time. So in other words, it's certainly going to be adaptations, but special meaning, what's on the market, forget about bacteria and things like that. But if you have something that's sitting, fighting against the immune systems of 30, 40, 50, 60 different species of animal, and the lifetime of the animal, let's call it, then you've got to have, that's a lot of evolutionary pressure. You've got to come up with some nifty response.
So what's interesting in this context, we should have systems that currently have eaten up for whatever the possibility to have this kind of applications. It's systems like neurosphere, where you have system that just hypermutates anything that's not mutated in the genome. Therefore, you're basically suicide if you try to copy a gene. So it's interesting to understand what are the evolutionary circumstances that seem to work a system like this. Is probably a genome gene system, as the logic interpretation for that is where everything that invades from the outside as a virus that integrates into the DNA receptor integrates multiple times. So if I can detect stuff that's using multiple copies, and I just get rid of this, I'm probably not hurting myself, I'm just taking new this. Versus a system that's very, very easily and very generally gene applications like animal genomes. It's very interesting to see where we are the different sort of external pressures of what are the circumstances where you can get one lineage really going into the lineage and say, okay, so I give up forever on the possibility to accommodate.
Yeah, I think it's a question of different niches, right?
Yeah.
I mean, it's a question of what variable would you use, characterize worldwide niches. I mean, same thing. So, many ways to skin a cat, right? So, many ways to survive. And the other thing is that, it's like, so, it's almost like many of the things there say the same thing.